right type. Blood type.
All right, here we are, ladies and gentlemen. Once again, let's keep talking about blood, and we'll pick up with blood types, where where we left off from the last video. Okay, whatever. So blood types are determined by surface antigens. Antigens are substances on the plasma membranes that basically tell the immune system, hey, I belong here. It's like the ID card, you know. You're walking around school with your ID card, your ID badge on you. That's proof that you're supposed to be here, right? Normal cells are ignored by the immune system. Foreign cells, which are cells that have antigens that the immune system doesn't recognize, are attacked. And so that's basically how your body recognizes that, like a bacteria or something like that, or a parasite is in your body that's not supposed to be. Your blood type is determined by the antigens that are present or absent on the surfaces of your red blood cells. Now, there are 4 antigens, A, B, and something called RH. It's maybe also referred to as D. We're going to use RH in this particular setup. And so we end up with 4 main blood types. Now, I will say as a disclaimer, there are way more antigens than just the ones that we're talking about, and blood typing gets really, really complicated, and then especially if you're doing organ donation and stuff like that, it gets even worse. But we're going to be talking about the 4 main blood types, and then there's like sub blood types in there, so there's 8 technically blood types. I'm getting ahead of myself. And so basically we have A, B, AB, and O. And so how it ends up working is blood type A has the A antigen. Blood type B has the B antigen. Okay, so far so good. AB blood has both antigens, some A's and some B's. Type O, if you notice, has no antigen. So that's how we can end up with 4 blood types out of just 2 antigens. Okay. Now, the RH one that I was telling you about is basically your blood type is determined by the presence or absence. There's not like ARHs and BRHs and stuff like that. So either you have it or you don't. If you do have it, then you are RH positive. So like type O, for example, would have no A's, no B's, but yes, RHs. So it's RH positive, okay? RH negative does not have the RH antigen, so a blood cell that has 0 antigens on it whatsoever would be O negative. We've got in your bloodstream, you've got special types of antibodies also, but I'm getting ahead of myself, and I apologize. So agglutinogens that's the name of those surface antigens, the A, the B, and the RH. Okay, these are what's screened by the immune system. Agglutinins are also known as antibodies, and these are the ones that are looking for antigens from foreign red blood cells. Okay, and they cause a process called agglutination of these foreign cells, also known as clumping. If you've ever seen milk curdling, it's kind of like that. Okay, and so basically we've got. So how it ends up working out then, if you have type A blood, you have A antigens on your red blood cells. You have anti-B antibodies floating around in your blood, which are looking for B antigens on blood cells. Okay? If you have type B blood, it's backwards. You have B antigens and anti-A antibodies floating around looking for type A antigens. If you have AB blood, you have both antigens. And if you look right here, you have no antibodies, which kind of makes sense because if you have both antigens, any antibodies for either one of those is going to mess up your own day, right? And so if you are AB, you have both antigens on your red blood cells, you are completely lacking in the antibodies looking for these red blood cells. Okay, and then the last one is type O. Type O has no surface antigens, and it has both anti-A and anti-B antibodies. Okay, and then as far as the Rh is concerned, only if you are sensitized to Rh. So basically, okay, so how it works, if you are Rh positive, you don't have, there's no Rh antigens. Rh negative is going to have Rh antigens, okay? Because it doesn't, the cells that it's trying to protect have no antigens on them. And I just realized I need to fix something real quick because I wrote that wrong. So let me go back and fix that real quick. Its antibodies are floating around. So Rh negative is lacking Rh antigens. It has Rh antibodies. Okay. And so that gives us, it kind of makes things difficult. I can't just give you my blood. I don't know, maybe I can. The problem is we can have a cross reaction or a transfusion reaction, which happens if you have a transfusion of blood from one person to another. If they are incompatible, then the antibody meets the specific antigen and the blood cells agglutinate. They clump together. Think of that curdled milk sort of thing, with the milk, the solid parts of the milk separating from the liquid parts of the milk. That's happening inside of your blood vessels, which is really, really bad news. Okay, and it can also lead to hemolysis, which is the breaking of red blood cells. So if somebody gives you blood that's not compatible with your blood, that can be a really, really bad thing health-wise. Okay, and so you can do simple blood typing tests. So basically, if you've got three serums. Okay, I've got an anti-A serum, an anti-B serum, and an anti-Rh serum. And if you see clumping anywhere, then you know that blood type has those antigens. Okay, so like in this first one, if we look, I've got clumping in the anti-A and clumping in the anti-Rh. That means that my blood cells have A antigens, so they are type A, and they do have Rh antigens, which makes them positive. Okay, same thing happens with B. AB positive, you're going to have clumping everywhere. If it's AB negative, you would not have clumping here. O negative is no clumping whatsoever. O positive, you would have clumping right here. Okay, so you do these simple tests for these three factors to determine if the blood is compatible or not. Like, I mean, it's more complicated than that, but this gets the job done. And so this is the cross matching that we would do to make sure that the blood sample the blood that we have for donation matches what is, the recipient, basically. Now there's sometimes you'll hear terms like universal donor. Universal donors are O negative because there are no antigens whatsoever on any of the blood, and so there's nothing for the antibodies to agglutinate with, and so O negative can be given to everybody, and so O negative is sought after a lot. Universal recipient would be AB because the blood's not going to react. The antibodies are not going to react to the antigens because there's antigens. The blood, the antigens in your body don't react to the antibodies in your blood do not react with the antigens on the red blood cells. And so if you are AB, you can receive from everybody, but donating is really tricky. A person who's O negative can only receive from somebody who's O negative because they have all the antibodies looking for everything. So it makes things kind of complicated. And like I said, these aren't the only factors that we're looking at. And so there is still, there's greater cross-matching than this that has to take place. So different blood types. Take a second. I kind of talked about this already in the last slide, and that's my bad. So take a moment, look at this. It sort of breaks down what I was trying to talk about and stuff like that. And you can also look at down here for the distribution of blood types in humans. So pause the video, take a look at this, jot down some information, and then we'll move on. Okay, so here's another one. If you haven't pulled the lecture notes that I've attached in Echo that goes along with all of this stuff, definitely do that. Pause and do that right now. This is a very handy chart that is useful as far as where the different formed elements come from and stuff like that. We didn't talk about the white blood cell ones. We'll talk about that in greater detail when we do the immune system and stuff like that. We focused on red blood cells, and now we're actually going to move into platelets. So platelets are also known as thrombocytes. These are cell fragments that are really important in the clotting system. They circulate for about 9 to 12 days, and then they deteriorate and are recycled. We make new ones. They're removed from phagocytes, primarily in the spleen. There's a lot of them, 150,000 to 500,000 per microliter of blood. A third of the platelets are stored in organs like the spleen and are only utilized during major circulatory crises, i.e. open wounds. Okay. Platelets release important clotting chemicals. They temporarily patch damaged vessel walls, and they reduce the size of the break and allow for the cells to regenerate. We have thrombocytopoiesis. This is the formation of platelets that occurs in the red bone marrow. They are formed by cells called megakaryocytes. Megakaryocytes are giant cells that are only in the red bone marrow, and basically they break off sections of themselves, little membrane enclosed packets of cytoplasm, and they just shed, kind of like we shedding skin, basically. And then from the red bone marrow, they circulate around, and then they eventually make their way over to the spleen. So here's kind of an overview of hemostasis. Hemostasis is the stopping of bleeding once some sort of damage to a blood vessel has taken place. And this is any size blood vessel, okay? Capillaries, smaller vessels, larger vessels, stuff like that. And there's 3 phases. There's the vascular phase, the platelet phase, and the coagulation phase. Now, I would recommend, if you're taking your notes digitally, take this picture off of the notes slide and drop it in your notes. This is a sort of overview of the different steps. We're going to go over them in greater detail right now. The first phase is the vascular phase, which lasts for about 30 minutes after an injury occurs. And so some sort of cut will trigger a vascular spasm. And so basically the smooth muscle fibers of the vessel wall will contract, narrowing the blood vessel. This lasts for about 30 minutes, which allows the other process to get started. In the platelet phase, the platelets begin to attach to the exposed surfaces. That's known as platelet adhesion. Okay. Then we have platelet aggregation, where the different platelets start to stick to each other, which happens pretty quickly, about 15 seconds after the injury is registered. And it forms a plug that closes small breaks. So in this phase, we have activated platelets are releasing a lot of different clotting compounds. ADP, serotonin, clotting factors, PDGF, calcium ion, stuff like that. So these are all chemical signals that are going out to the rest of the world. Okay, and so if the plug can't be formed by just the platelets, then we get into the coagulation phase. Now, things that limit the growth of the plug. It does happen naturally. Sometimes you don't necessarily want these to occur. We have different hormones that inhibit aggregation. White blood cells will stop the platelet plugging process. Enzymes break down ADP, which is going to slow it down. Negative feedbacks and the blood clot isolates area from general circulation, basically meaning the clot is going to close the area off, basically. So we get into the third phase, which is the coagulation phase, which happens 30 seconds or so after injury, which is going to depend on the concentration of clotting factors that are released, like calcium and some of the other ones that we had talked about. They're going to start out as proenzymes, which are just inactivated enzymes, and then they are converted to active enzymes that are when they come in contact with the clot that is forming. And so basically what we end up with is this. It's a chain reaction of different pathways. We've got the extrinsic pathway, which is happening outside of the circulatory system. So like within the tissue itself, for example. And then the intrinsic pathway, which is taking place within the blood vessels. Okay, and so basically what ends up happening is these different pathways release clotting factors. Now, I'm not going to go into a whole lot of detail about these. Take some time, look at this chart, copy it down into your notes. But both of these pathways, basically releasing different types of factors, end up going into a common pathway where we have something called clotting factor 10 or clotting factor X. These stimulate the release of a substance known as thrombin. Thrombin is going to start a positive feedback loop. So thrombin is going to convert fibrinogen, remember we talked about that, one of the blood proteins, into fibrin. Fibrin is basically going to create a network of, fibers, kind of like a net, that are going to trap the platelets and then the red blood cells, and that is a blood clot. As the thrombin is being produced, it is going to trigger the release of tissue factor and platelet factor, PF3, to ultimately produce more thrombin, which is going to produce more of the other factors, so on and so forth, until eventually it stops when the clot is completely formed and has closed off the hole. Other things that control blood clotting as far as slowing it down and stuff like that are anticoagulants like antithrombin 3, heparin, which is another substance that activates antithrombin 3, thrombomodulin, which and prostacyclin all can inhibit the formations of clotting through different pathways and stuff like that. Ions that are super important, calcium and vitamin K are really important in the blood clotting process. All three, the extrinsic, the intrinsic, and the common pathways, all require calcium. So here's another reason why calcium is super duper important. It's not just muscles. Vitamin K is required for synthesizing 4 of the different clotting factors. Let's see. Disorders of bleeding and or clotting include thrombocytopenia, which is low platelet level, so the clot doesn't form. Hemophilia is a lack of clotting factor, so the clot doesn't form. Thrombophilia is the clots form too easily because basically you have too many platelets or too much of these clotting factor proteins. Deep vein thrombosis is a clot that forms in a deep vein, typically in a thigh or lower leg, but it can happen in other blood vessels besides that. And those are can cause a lot of problems, movement problems. The clots can break apart and go to other parts of the body and stuff like that. The final step that was on that first diagram right at the very beginning is clot retraction. And so basically as the clot forms, it's going to pull the edges of the vessel closer together, which is going to help control bleeding and reduce the size of the injury site. It's also going to make it easier for fibroblasts and muscle cells and stuff like that to begin regeneration and repairs. As far as breaking the clot down, that's known as fibrinolysis, which is the clot dissolving, driven by thrombin and tissue plasminogen activator, which is going to activate the plasminogen, turning it into plasmin. The plasmin are going to digest the fibrin strands, and then the clot is going to go away. Here is that chart again, if you didn't catch it from the last one. Pause it, take a look at this, maybe draw some notes in your chart, or take this picture and put it into your notes if you're doing it digitally. Well, shoot, ladies and gentlemen, that brings us to the end. So what did we talk about in the last two videos? We talked about the function of blood. We talked about the components of blood, including plasma and the formed components. Remember, the formed components are the the red blood cells, the white blood cells, and the platelets. We talked about human blood typing and how which antigens will determine your blood type. And we're actually going to practice with some of that the next time I see y'all. Later in the week, we've got a case study that we're going to look at. We talked about red blood cell formation, erythropoiesis. We talked about hemostasis. What does your body go through in order to close up those wounds so you don't bleed to death and all that other stuff? Let's see. If you have any questions, let me know. Send me a remind message, send me an email, ask me in class. Take a break from this one. We do have another video talking about the anatomy of the heart that you should probably take a look at before we get into our heart dissection, so you know what some of the stuff is that we're going to be looking for. I guess that's it. I look forward to seeing y'all again soon.
Nobody makes me bleed my own